Hello, I'm Jill Marzolino, a PhD student studying under Dr. Dan Koenig of the Botany Department at the University of California at Riverside. The EPPN 2020 grant sponsored our research project, The Phenotypic Basis of Barley, PHENOHORD, Success in a 90-Year-Long Competition Experiment. Thanks to this grant, the Koenig Lab was able to send Dr. Jacob Landis to the IPK in Germany to acquire high-throughput phenotyping with their LTA and LTC systems. This research was made possible with the help of Thomas Altman, Astrid Juncker, Julianne Streit, and Henning Schierch, as well as Claude Becker at the Gregor Mendel Institute. My research focuses on environmental adaptation in crop plants. I'm interested in this topic because of the threat and challenges posed to global food security because of climate change. Plants are dependent on their environment for survival, and, since they can't find a better home like animals, have to cope with their surroundings. Climate models predict strong changes in temperature and precipitation, two of the most important factors in successful plant survival and crop production. I use barley as a study system because it is the fourth most important grain crop in the world, and can be quite drought tolerant, and has diverse genetic resources from barley landrace populations growing in varied environments around the world. Furthermore, barley has been used to found a really unique experimental evolution population that lets us examine the genetic process of adaptation over time. The composite cross II population was synthesized in 1929 by Harry Harlan and Mary Martini by crossing 28 landrace parents drawn from major barley-growing areas of the world, pooling their progeny, and growing subsequently produced seed in an agricultural setting in Davis, California, without conscious selection for over 100 years. Seed is kept from each generation, preserving each step in the process of evolution, and allowing us to study the process of adaptation to the warm and dry Davis environment through time. In the composite cross population, we've historically observed three strong phenotypic shifts, including an increase in yield, flowering time, and height. These phenotypic shifts are exciting evidence of evolution and match up our observations of a strong reduction in genetic diversity over the generations. Unfortunately, though, manual phenotyping has limited our observations to a handful of traits at the end of a plant's life cycle. This means there's no temporal resolution. We have no records of how plants grow and whether developmental patterns have shifted during adaptation. With more comprehensive phenotyping, we may be able to better describe adaptation to this particular environmental niche. To do so, we have initiated an experiment to collect data on over 400 traits for every day of a plant's life. This international collaboration allowed our postdoctoral researcher, Jacob Landis, to travel to the Leibniz Institute of Plant Genetics and Crop Plant Research in Gatersleben, Germany, to plant and run a large-scale phenotyping project on our composite cross population. To uncover the changes in growth patterns and development, 164 progeny lines from 4 generations, plus the 28 founding parents of the composite cross, were planted on 2 large-scale phenotyping machines at the Institute. One machine carried seedlings for 2 weeks, and then a larger machine grew and phenotyped the larger maturing plants. Over the plants' entire lives, every day of growth was captured for 401 traits, and photosynthetic capacity was recorded weekly. This daily resolution allows us to document the growth rates and developmental patterns of individual plant lineages and generations of the population as a whole. Plant phenotypes can now be paired with previously collected genome sequences to link phenotype to genotype. By comparing the range of trait values and allele frequencies across and within the generations, I can determine how the environment shaped this population, how selection acted over time, and which traits are most important for successful adaptation under the